Welcome back to botany class. Let's look at a glance of today's learning objectives. Let's start from biofuel or algal biofuel, hydrogen production by alga, bioprospecting including biopiracy. Here you are going to learn three different types: neem, turmeric and basmati rice, and applications of biotechnology. 4 5 more question you are going to learn today. Biofuel or algal biofuel, hydrogen production, bioprospecting including biopiracy, applications of biotechnology. Let's see the first question. Biofuel. Biofuel or algal biofuel or algal oil is the alternative source of biofuels like petroleum products, fossil biofuels or liquid fossil fuels. So, it is the alternative source. This algal biofuel, biologically fuel is obtained from alga. So, algal biofuel. Normally biofuel, the source of biofuel is sugarcane or corn, but this from maize or sugarcane it is obtained, the biofuel is obtained. Here instead of the liquid fossil fuels, the petroleum products or fossil fuels, alternative source of these fossil fuels is algal biofuel. So, these biofuels are obtained from algae. You know that there are number of classifications of alga. Fritz classified 11 different classes. Last year you learned these classifications and its type of pigmentation and so on. But here in this chapter you learned how the biomass production using the bioreactor or fermenter by fermentation. So, here the algal culture, the energy crisis and the food crisis initiated the culture of alga. So, here the alga is cultured for obtaining the fuel. So, look at this diagram here. The most important alga or the widely used alga for biofuel production used as Botryococcus brownie. One word question which is very important. Botryococcus brownie is the alga widely used for the production of this biofuel. So, biofuel which is otherwise called algal biofuel. Algal biofuel is the fuel which is obtained from algae. How to obtain this alga? So, to obtaining oil or this biofuel, so it's this alga not only gives the biofuel but also it gives energy rich oils. Energy rich oils also obtained from this. Also you studied already the single cell protein SCP, protein contents also taken from this. So, using of this one Botryococcus brownie, there are several kinds of alga used to prepare single cell protein. The economic importance of alga is, alga plays very important role in the economically it plays very important role. But here Botryococcus brownie is the alga widely used to produce this biofuel. So, at first the alga can be collected. So, different kinds of alga. So, this variety of alga, that is Botryococcus brownie, is collected. You know the alga which is freshwater as well as marine water. So, this alga contains lot of contaminants. So, at first it can be washed it or purify it, and different containers, the bioreactor, there are different containers used to culture the alga. It may be unicellular, so it has to culture abundant to obtain more a number or more. So, this alga can be cultured in different containers. The containers are sterilized, and the needles, what are the things used for transferring this, make it sterilized. All the equipments, all the hardwares, all the vessels can be sterilized. So, culture, at first it can be culture, algal culture. There are different colonies of alga which is culture in different containers. It takes the sunlight because it is a green alga, most of the alga which performs photosynthesis. So, it obtained this energy, source of energy, sunlight, and it synthesizes photosynthetically. It takes carbon dioxide and it releases oxygen, but it respires and releases carbon dioxide. And this culturing, when it is cultured, the final procedure of culturing is purification. Why it is culturing? To obtain different product. So, from alga there are different products can be obtained. Here there are three important products can be produced. The main product is biofuel. This is the alternate way of getting biofuel. So, first oil extraction. Oil can be extracted, and the protein content. So many alga which is rich in protein. So, protein also separated, the protein residues are separated, and the biodiesel and energy rich oil. So, three different kinds of products can be isolated from this algae. So, the oil is extracted, oil extraction, and the protein residues can be separated out from the oil. Also, it produces the biodiesel, the production of biodiesel. So, this biodiesel, the renewable fuels, it produces renewable fuel as well as glycerin also obtained. So, this can be separated, and the renewable fuels are used in the existing technology. So, it is used for the the renewable fuels are used to the vehicles, and this vehicles, from the vehicles it releases carbon dioxide. So, this biofuel can be extracted from the alga. Very simple, but for telling it is very simple, but culture it, there are different process and it can be separated. Final process of culturing is purification and separation of different products. In this way they are obtaining this biofuel from alga. So, it is called as algal biofuel. Next question is hydrogen production by alga. So, hydrogen is produced by alga. So, this technology is normal photosynthetic technology. You know that alga, which is it is a green alga, green chlorophyll pigment is there. So, photosynthesis taking place. So, this photosynthesis, there are different kinds of photosystems, PS1 and PS2. So, normally this production of hydrogen is the process by which when it here the water molecules, splitting of photosynthetic water molecules. So, photo splitting water molecule, by this method hydrogen produces. When it produces hydrogen, when it deprives of sulfur, it produces hydrogen. Normal photosynthesis, here chlamydomonas, you know this alga, chlamydomonas, which produce, this undergo photosynthetic process and it releases oxygen. Normally when the photosynthetic process is going on, it absorbs all the carbon dioxide, it takes carbon dioxide and it releases oxygen, it evolved oxygen. So, here in this photosystem PS2, the electrons captured, the electrons are passed through different electron carriers. The electrons obtained from PS2, it passed through different electron carriers. What are the electron carriers? Here PQ, plastoquinone, cytochrome B6, cytochrome F and plastocyanin. So, these all electron carriers which passes the electron from PS2 to PS1. Then finally, here during this process of normal photosynthesis, splitting of water molecules taking place, because by photosynthetic process water, 2 molecules of water, 2H2O. So, it splits into, these 2 water molecules splits into oxygen and this 4 protons. It splits into protons and electrons. The number of protons are equal to number of electrons. So, 4H plus 4E minus. So, this water molecules, 2 water molecules split into protons, electrons, and it evolves oxygen. During photosynthesis, oxygen is evolved by splitting, photolysis. So, here this process, water is splitting into protons and electrons. 4 protons. So, number of protons are equal to number of electrons, and this oxygen is released. Now, this electron which is passing through PS1 is captured by FD, ferredoxin. In normal photosynthesis, what happens? This carbon dioxide fixation by RUBP, carbon dioxide fixation. So, here it is FNR, that is ferredoxin NADP plus reductase. Ferredoxin NADP plus reductase. So, it is used for carbon fixation, but for the production of this hydrogen, here the question is hydrogen production by alga. Hydrogen production by alga. So, when it produces hydrogen, when this sulfur is deprived, if deprived of sulfur, these electrons pass through different electron carriers from PS2 to PS1, and this is captured by ferredoxin, FD, ferredoxin. So, this ferredoxin, from here H2AS enzyme, that is hydrogenase enzyme. Hydrogenase enzyme combines with ferredoxin and it produces hydrogen. So, hydrogen production, very simple. This ferredoxin captured electron. Here there is an enzyme, hydrogenase enzyme. Hydrogenase enzyme combines and it produces hydrogen. So, ferredoxin combines with hydrogenase enzyme and it produces hydrogen. So, by alga hydrogen is produced. When it produces, when sulfur is deprived, hydrogen is produced. This is the second question. Then another question is bioprospecting. Bioprospecting is the process by which production of new products from the indigenous knowledge obtaining from the indigenous plants or indigenous resources. From indigenous resources, they are producing a new product which includes or involves biopiracy. So, here is the production of new product and commercialization is bioprospecting, but it involves biopiracy. Biopiracy is the process by which there are number of indigenous resources, indigenous natural resources are present in so many countries, but these resources are not used by the indigenous people, but it can be taken from some other people or some other countries without the proper authorization of the indigenous people. That is called biopiracy. So, 3 examples we are going to learn, that is this neem, turmeric and basmati rice. So, this neem, turmeric and basmati rice, first let us see one by one. Neem, we all know that neem and turmeric are used by Indians. So, it is the subcontinent. It's the nature of Indo-Pak subcontinent. So Indians, they are using this neem and turmeric as the medicinal plant. So this neem, let's take about this neem. The neem, we are using the neem plant parts and the neem oil for controlling the fungal and bacterial infections. So Indians, they are using this neem plants leaf as well as the neem oil for controlling fungal and bacterial infections. So it's not a new product, it can be already known because the people normally, traditionally they are using this, but it can be get a patent to multinational companies. For example, in United States, in US, the patents can be given for this neem oil. So the multinational corporations, they give the trademarks or the patents can be given to them. So without proper authorization of this indigenous people, they are using this with patent. So this is one example of biopiracy. Another example is turmeric. We know that the value of turmeric. In our houses, we are using this turmeric as an antiseptic agent. Antiseptic. So every houses it is used as an antiseptic and quickly it heals the wound as well as it heals the rashes. So it's used as an antiseptic. It's not a new product, it's not new. So this term, the Indian Medical Council, it already published a journal to the world and they explained about the medicinal value of this turmeric. So the use of this turmeric, so it's not new to the world, but it also patent to the companies, multinational companies and the trademarks. So this neem and turmeric are one of the example of biopiracy. Another important example of biopiracy is basmati rice. In the year 1997, September 2nd of 1997, the US patent trademark give the basmati rice, give patent or grant patent to basmati rice lines and grains. So basmati is the native of or it's the indigenous plant of Indo-Pak subcontinent. So this basmati rice, they get patent to US company. So US patent trademarks gives authorization over this basmati rice as well as grains. Also they give this Texas based company, Rice Tech, the technologies they are using. Actually these all plants are the traditionally used plants by India, but they are getting authorization over this indigenous plants and they are getting patents. So patents have been given for rice technology, breeding technology regarding seed as well as the grains. So also they give breeding technology, they are using the breeding technology rice techs, novel rice lines. So all these technologies can be used by another multinational corporations. So that this has been taken to WTO, World Trade Organization. So the people of this traditional plant or indigenous people, they take it to WTO, World Trade Organization, that is TRIPS. TRIPS means trade related aspects of intellectual property. So this trade related aspects of intellectual property, So they take this to this TRIPS. So it was the embarrassment to the US people. Hence, in the year, so they dropped certain claims. They dropped. So certain decisions they took, and this rice technology, they dropped certain claims. In the year 2002, this rice tech, they can develop so many varieties, so many products they can produce from this source, from this grain, seed, and different technologies of breeding technologies can be used, and they can sell it without authorization of this indigenous people. So this regarding basmati rice is an embarrassment to the US, and in the year 2002 this rice tech dropped 15 claims about this basmati rice. So the patent name also changed as Rice Lines 867. Now this patent name has been changed as Rice Lines 867. At first, the Texas based company, it gives the rice technology. They give full authorization to this rice technology, rice tech, novel rice lines. They can breed and they can use the grains, seeds, so many products they can produce. But now in the year, the final decision can be has been taken in the year 2002, and they dropped 15 claims and changed the patent name as Rice Lines 867. So this is one question you are going to learn today. Then finally there is a question: applications of biotechnology. What are the applications of biotechnology? So this in this chapter biotechnology you are going to learn from the very first class itself. Biotechnology is a interdisciplinary sciences of this century, 21st century. So it's the interdisciplinary science. The biotechnology is the interdisciplinary sciences of 21st century because it gives a way to apply the products, getting a product or producing a product, and applicable, it can be applicable to the mankind and the animal resources. So this biotechnology, so many technologies are applied for the use of life processes, life to mankind as well as the animal or the cattle. Then in biotechnology, what are the applications? So first application is, it's the interdisciplinary sciences. Interdisciplinary science, and it includes number of sectors. There are number of sectors including this biotechnology. What are the sectors? It like agriculture, medicine, environment, and industries, commercialization. So many fields, agriculture, there are so many new technologies can be used. Applying different technologies to develop new qualities or different fields, medicines, vaccines, antibiotics, medicines, this all produces under medicine. Then agriculture, in agriculture there are so many different areas. This technology, biotechnology can be implemented and produces new qualities as well as quantities of different products. Then transgenic plants. Transgenic plants. You learned about transgenic plants or genetically modified plants, GMO, genetically modified organisms. Plants also modified, or this organisms also can be modified. So by transgenic plants, they are producing disease resistant, pest resistant varieties, viral resistance of so many plants like tomato, potato, banana, so many. Also, tissue culture technology can be used. Under tissue culture, the protoplasmic fusion or the single cell culture, including the SCP production, single cell protein. Here we studied that biofuel, algal fuel, the natural resources all obtained by this transgenic plants. Insect resistant, disease resistant, pest resistant varieties, saline resistant, saline tolerant and stress resistant. So many varieties can be obtained through this transgenic plants, and it gives an immense outcome of biotechnology. It produces lot of outcomes through these all technologies. So under this medicines play a very important role in this 21st century. This medicines are very important. One of the production deficiency of insulin, production of insulin. If the vaccines or if the products, insulin, the hormones or plant growth hormones, or here the human insulin can be produced or proteins can be obtained by the using of E. coli bacteria. So bacteria plays very important role. It used as a vectors, as a cloning vehicle. The widely used cloning vehicle is E. coli, Escherichia coli. So one of the example, very important example is production of insulin production using this vector E. coli. And nowadays the biochip, production of this biochip plays very important role in the field of biotechnology and biology. This is the biological computer biochip to detect all the organisms, several animals or even so many organisms used by this biochip. And genetic engineering or this genetic manipulation or altering the gene. So many examples you studied under this chapter earlier. There are number of varieties can be obtained, cloning method, altering the genes, manipulation of gene. A desirable gene can be isolated from one organism and introduced to the another organism as our choice and obtain a new variety or new product. Also secondary metabolites. So there are number of metabolites. Commonly there are number of metabolites are present in the plants in different parts. It can be isolated and produces the secondary metabolites in the form of liquids, oils, resins, gums. This all produces by this. And this one finally single cell protein. Then biofuels, bioremediation, and this all hydrogen production, biofuel production. This all technologies or new technology is used, and all these technologies are applied to the improvement of producing different products. That's the application of this biotechnology. So it's one of the important question, 5 mark question. So once again, let's see or review what you learned today. First question is biofuel or algal fuel. Draw this flow chart and explain this biofuel production, and draw this diagram and write hydrogen production by algae. Then third question, bioprospecting, write the definition. Involving biopiracy, write the definition, and 3 examples are there: neem, turmeric and basmati. Then last question, applications of biotechnology. So up to this applications of biotechnology, fourth chapter completed. In the next class we will meet with chapter 5. Thank you.